Thanks.
Thank you, and I want to thank you for the invitation to talk at this meeting. This is a great meeting. It's always a lot of fun to come out here. It's particularly cool for me to give this talk today because I'm the only one speaking who's representing the 3 bioenergy research centers. So what I thought I would do in my talk is first go through Great Lakes Bioenergy as an example of what DOE is trying to do with the bioenergy research centers very quickly, and then spend most of my time dealing with the major topic of that was in my seminar title. So let me first introduce you to Great Lakes Bioenergy. So Great Lakes Bioenergy is, as was said, one of the 3 DOE bioenergy research centers. It was established a little over 8 years ago now. We're in our 9th year. It's housed at UW-Madison. It's anchored by UW-Madison and Michigan State University, but it's actually an international team of researchers at 7 universities and a national lab in the US and Canada. It includes about 450 students, scientists, and staff, all with a common mission of generating knowledge to improve the sustainable production of crops with desirable biofuel traits, and to improve our ability to have systems that allow for the energy-efficient conversion of lignocellulosic, or the non-edible part of plant biomass, into fuels and chemicals. So there's a large genomic component in this, but given the way we are structured and the amount of support that we get from DOE, we've been able to address this problem by building up, if you will, an integrated biomass to biofuels and chemicals pipeline, much like in an academic setting, much like what a company would do as they take feedstock in the door, pre-treat it, convert it into, we call them hydrolysates. If you were in the wine industry in Napa, these would be your juices that you're going to ferment into fuels and chemicals. And then analyze the material as it flows through this pipeline and look at the outputs. Everything from genomics to rhizosphere biology and fermentation work. So in part of this, you can look at it this way, and you can see that we have activities across our many sites. And here's JGI as a key component of this that provides us with a unique resource for gene sequencing as well as gene synthesis. And both of those activities will be highlighted as I go through my slides today. This is one way of looking at this pipeline. The other way of looking at it is depicted on this slide, where we can see we have people in the center that work in the field, either on our own at experimental stations in the state of Wisconsin or at Michigan State University, but also more and more we want to work with people who are looking at experimental stations at other places, and maybe our next speaker will have something to say about that. We then treat that biomass, turn it into these juices, and ferment it. And one thing that goes on in all of this is the production and the analysis of large and disparate data sets. So we take all of that data into the center, we store it, we make it accessible to investigators, and then we have the ability to have a microbiologist like me at the right-hand side of this chain looking at a fermentation, and all of a sudden something is awry compared to what happened the last 2 times we did it. We can go back and see where that crop was grown, what the growing season was, what the weather was, and how it was processed, and see if there are similarities or differences to inform scientific discoveries. So it's a large integrated set of activities in the center. What I'm going to do mostly today, though, is talk about some of the pieces that we have been able to put together to address individual problems, and then in the end I'm going to come back and try and tie them all together. So moving on to the mining genomes for fuels and chemicals, the first example of us really doing something that I think was innovative and that was enabled by JGI was some work that we did to improve the traits of biomass plants for processing into fuels and chemicals. Society has done a great job in improving crops for food, but but we've done nothing to really think about how we could improve them for fuel and chemicals and also allow their continued use for fuel. So what I'm going to do in the next section is give you one example of the types of things we've been able to do. So one of the major goals of the program when it was started by DOE was to enable the discoveries needed to rationally change plant cell walls in order to improve biomass processing. We know from work in the literature and others way before us that a lot of the carbon that we want to get at in these plant cell walls is tied up in two types of major polymers, polysaccharides and lignin. And in order to get at those and make fuels and chemicals from them, we need to be able to separate them easier and get them in a fashion that's going to be amenable to downstream processing in the valuable products. So the example I'm going to give where we've been able to actually go in and rationally make changes to plant cell walls is a set of experiments where we took upon ourselves the goal to go in and redesign the lignin polymer in the plant cell wall background. And in order to do this, we wanted to go in and modify the lignin, not to get rid of it, but to make changes in it so that it would be easier to break apart into its component pieces. And specifically, the approach that we wanted to take was to go in and put in chemically labeled ester linkages in the lignin polymer. So these two cartoons, in a not very chemical way, describe the lignin polymer. It's built up of a series of aromatic rings that are formed, that are covalently attached to each other. The different colors of the rings represent the fact that they are different aromatic substituents. And this approach that we wanted to take was to go in and put in these twist ties to make it easier to separate those rings when you were going to process them. And the upshot of that approach would be what you see down here in this structural model of the lignin polymer. Everywhere we put one of those twist ties would be this green bond in the lignin. And now with mild chemical and treatments, it would be easier to break apart. So for every cleavable bond you put in, you would get 2 new products out. So how could we do this? The approach that we took was based on the fact that we knew there were examples in the botanical literature of plant cells that have high contents of these ester linkages in their lignin. And this shows our favorite model, Angelica sinensis, which is a Chinese medicinal root, has almost 90% ester linkages in its lignin. And so very early on, we did some, I think, elegant experiments with the JGI's help to do tissue-specific RNA-seq from this plant and identify genes that were differentially expressed in those samples as a way of identifying candidates for a monotransferase that would provide the monomers that the plant would then put together to make this polymer that has these twist ties in them. So we identified candidates, confirmed their activity of those gene products biochemically in yeast and in Arabidopsis, and have now actually moved on and put them in poplar. And the next slide shows that when we put these materials in poplar, they go to the right place in the plants. And when you process that material, this slide, you get improved release of sugars from the zip poplar, as we call it. So the control here shows the release of glucose and xylose, 2 of the major sugar monomers from a wild-type poplar line. And here is a set of lines comparing how easy it is to get the sugar out when you have this zip trait in this poplar strain. We've set up assays that didn't exist when we started at a 2D NMR. I won't bore you. If I show you a bunch of spectra, I'll probably put you to sleep. That showed that the zip trait is incorporated in the lignin and it's incorporated at both ends. So This is a clear example of using genome-enabled gene discovery and being able to modify cell walls to improve biomass processing. So what we have shown is that modifying lignin biosynthesis gives us walls that are easier and cheaper to process because it can now be done at lower temperature with less harsh chemicals. And we now that we have these assays, which didn't exist, we actually see these esters spread throughout the plant kingdom much more widely than we thought they existed 8 years ago. So we're very interested in identifying, and we have candidate genes that would put these zips into native plants. We're very interested in leveraging the ability of native plants to make these esters as a way to improve biomass processing in a variety of crops. Everything I've shown you so far on the Zip Poplar project has been done with trees that are grown in nurseries. We actually are excited to have trees that are in the real soil in Wisconsin and Madison and in Michigan this year. So we're moving forward to bulk up a lot of material to see how these Zip trees work in nature and to also begin to address how well they function in making fuels and chemicals from this type of a substrate compared to wild-type plants. Moving on to my second example, I want to move down the chain here and talk for a few minutes about what we've been doing to make it easier and cheaper to try and make these juices or hydrolysates from biomass. And in particular, what I'm going to do is give you one example of work that we've done actually with JGI and JBay, one of the other centers, which is right across town from here, to identify improved enzymes that will take these polysaccharides apart into their component sugars. The approach that we've taken to do this is to look at naturally cellulolytic ecosystems. And over the years, us and the other two centers have screened a lot of native ecosystems that have high cellulose degrading activity. And we made a decision early on to look at insect-associated microbes. And the story I'm going to tell you today comes from the analysis of streptomycetes that are associated with this wood-boring beetle. When we first looked at this ecosystem and began to enrich for organisms on the nose of this beetle that inserts itself into the wood, we found that they were a relatively simple ecosystem that were dominated by streptomycetes. And if you isolate those organisms and look at their supernatants, they have high cellulolytic activity, almost as high as the best enzymes that are sold by industry to process cellulolytic biomass. So with the help of JGI again, we have now gotten sequence from a lot of cellulolytic streptomycetes, and you can see that the insect-associated members of this clade of this tree fall in one particular clade. And every time we look at these secretomes, we see very, very simple enzyme patterns. I'm not going to show you all the data. So it led to the hypothesis that there was something different in the media that was secreted by these streptomycetes that allowed them to be much more robust in cellulose and hemicellulose degradation than the systems that had been studied before. So that was the good news. The bad news is we had a lot of isolates and we had a lot of candidate open reading frames to test that would have been very laborious, again, if it wasn't for JGI's help with rolling out their gene synthesis platform. So we actually went to JGI and were able to order up genes for many of the open reading frames for many of these insect-associated streptomycetes, literally hundreds, that were each cloned separately into a plasmid. We then took them back at Great Lakes Bioenergy and expressed them in a robot, in a cell-free system, in a wheat germ system. Instead of doing a laborious set of experiments where you would express them in E. coli and worry about folding and stability and all of the things that normally kill you, we made them in vitro, and then we set up this simple screen to test their activity on a variety of polysaccharide polymers. It's not important what the enzymes are on the y-axis or the substrates are on the x-axis. What's important is to look for colors. The typical enzyme in a screen like this works with one polymer reasonably well and not with too many others. Or maybe you get something that works with two and has wimpy activity. But here's an example of two enzymes from these insect-associated streptomycetes that gave us dark color across a variety of substrates, arguing that a single polypeptide chain was able to hydrolyze multiple polysaccharides, something that had never been seen before. And with the help of Trent Northern at JBay, we set up another step now to screen these enzymes in a high-throughput manner by mass spec using a NIM system that he put in place. And this allows us in real time to monitor polysaccharide degradation by each of these candidates and look at the individual products. Are they working from the end and giving us a monomer? Are they working as an endo carbohydrate hydrolyzing enzyme and giving us a polymer? And since we can do it in real time, we can actually work out enzyme kinetics. So the long and short of this is that we have identified a series of enzymes from genome mining from these insects that we now call CMX enzymes because they have the ability to cleave each of the major polysaccharide polymers that are found in plant cell walls: cellulose, mannose, and xylan. Cellulose, mannans, and xylan. That's why we call them CMX. And this shows over here the activity of a variety of these enzymes on different substrates, and wherever you see a bar across all three slots, that means it works on multiple polymers. Also here we have structures of candidates of each of these, and we are now using molecular dynamics to look and see what's different about the active site of these enzymes that fall into a part of a major clade of enzymes that previously have been reported to be specific either for cellulose, mannan, or xylan. So there's something different about these enzymes that we're very interested in. So to tie this up, we've been able to show that this new family in a large glycosyl hydrolase clade is able to hydrolyze multiple polysaccharides. And we've also gone on to fuse these domains to so-called carbohydrate binding domains that are found in other cellulolytic organisms that actually serve to bring the enzyme to the substrate. There's a binder and an arm, and then the enzyme goes as a Pac-Man and eats on it. And in some cases, we've been able to show that carbohydrate binding domains like this one, when put onto one of these CMX enzymes, actually leads to a large increase in enzyme-specific activity and turnover. So we're very excited about this because we think the properties of these enzymes, their broad specificity, could actually reduce the number of enzymes and therefore the cost of making enzymes that industry needs to use to process plant cell walls. The last story I want to tell you about is work that we've been doing to improve biofuel microbes in the center. And I'm not going to talk so much about the types of molecules that we're making in the center, but instead what I'm going to do is talk about, I think, what has been one of our unique approaches, and that is to see how we've been able to use genomics to understand the impact of biomass-derived toxins on biofuel microbes. So a lot of people in the field think that when you make these hydrolysates, it's just like juice that you're going to use to make wine. Well, it turns out it is a lot of sugar and it's a lot of food for most microbes, but it is also replete with a large amount of toxic molecules that come from processing plant biomass. And in order to help industry be more successful, we need to understand what those inhibitors are and begin to engineer processes around those inhibitors to improve either the plants or the microbes' ability to make fuels and chemicals. So again, enabling the studies that I'm about to tell you about was this platform of activity that I mentioned earlier, where we have set up systems to monitor carbohydrates, amino acids, minerals, metals, anions, and all sorts of other organic compounds as we process this biomass. This allows us to know, whenever we run a fermentation, the chemical identity and quantity of over 100 compounds in that media. It's like saying I know everything that's in LB. And in fact, we don't, but we know everything that's in these hydrolysates. And this shows you what's in these hydrolysates. You don't need to read it. I put it up because it's a long and impressive list. But this slide shows that we can now make synthetic hydrolysate using this chemical composition and go to the reagent shelf and recapitulate all of the negative effects either at the gene expression level or at the fermentation level with synthetic hydrolysates that we see with the real McCoy in this example from Kornstova. The bottom of the slide shows that we can actually do takeaway experiments now, where we can remove one compound or individual classes of compounds from this synthetic hydrolysate and ask which ones of them are having a negative effect on the biofuel microbes. And the rest of what I'm going to tell you about today are the lessons we've learned by identifying aromatics that come from cell wall degradation when we make these hydrolysates and how they impact negatively the performance of biofuel microbes. And there's a genome-enabled, sequence-enabled, strong bias in how we are going about doing this. So we're presented with a solution that has a variety of inhibitors in it. What do we do to go figure out what they are? What we have depended a lot on is using something called chemical genomics to get a genomic fingerprint of these so-called lignocellulosic toxins, LTs. And the way this system works is outlined on the top of this slide. We take a random collection of mutants in every gene in an organism of interest. We grow that collection either in standard media, in a hydrolysate, or in media where we've added one or more lignotoxins. After growth, we take that population and we sequence it. And we ask who's left from the starting population after the fermentation. And you get a so-called chemical genomic profile where most of the organisms track this way, if this is the number of genes in an organism. These blue dots down here are mutants that are sensitive to whatever that treatment is because they are lower abundant after the experiment than they were when they started. And the yellow ones are resistant mutants because there's more of them after the experiment than there was before. We've actually done a lot of this with 3 different biofuel microbes: yeast, Zymomonas mobilis, a well-known ethnologen, and E. coli. And what amazes me that's come out of this analysis is when you do these experiments with a large number of hydrolysate compounds on these 3 different organisms, you come back with a very simple answer of what these toxins are doing. Most of these toxins are blocking energy and nucleotide metabolism in all 3 of these organisms. So across a large evolutionary space, all of these lignotoxins are honing in on one part of metabolism. So let me give you now a couple of examples of what we've learned by analyzing this at more mechanistic detail. So one of the abundant lignotoxins is this compound here, a compound known as ferulamide. It's a ring with a side chain on it that comes from plant cell walls. And this growth curve down here shows you that as soon as you add ferulamide to an E. coli culture, growth ceases. But if you do flux balance assays, you see that label accumulates in this compound here, phosphoribosyl pyrophosphate, which if you go back to intermediary metabolism, is a key intermediate in nucleotide biosynthesis, and nucleotides are needed for energy metabolism. If you go further, you can see that as soon as you add the ferroamide, the ATP and CTP pools drop, and this explains why this compound is a toxin. We now have a target enzyme to reverse engineer around to look for mutants that bypass this toxicity. So that's an example of what we've done with one major lignitoxin. Here's an example of what we've learned by studying another one. This story is about this compound here, Poasic acid, which is actually an aromatic dimer. That's rather low abundance, but it's actually a very potent and specific inhibitor of yeast. The chemical genomics assays that I described a minute ago showed that its target, or predicted that its target, was in cell wall or beta-1,4-glucan synthesis. And this is a well-known target for pharmaceuticals as an antifungal. This shows that if we take Poasic acid, it actually protects soybeans from the toxic effect of a known fungal pathogen. So here now, we're very excited about this because we've actually been able to generate a valuable co-product, possibly for agricultural biotechnology and human or animal biotechnology as a byproduct from processing plant cell walls. So to summarize, we've been able to identify systems that are targets for biomass inhibitors that negatively impact biofuel microbes, and we've been able to increase tolerance to single inhibitors. We're now moving on to work more on designing biofuel microbes that will face the multiple inhibitors they see in these hydrolysates, and in an iterative way, working with our crop scientists to go and modify plants to try and mitigate the levels of these inhibitors that will be present in biofuel crops as we move forward. So in sum, I want to leave you with this slide that compares where we want to go with making products from things like corn and poplar and perennials to what's currently done in the oil industry to make value from a barrel of oil. On the left is a cartoon I stole from an EERE document, and it illustrates that the oil industry makes about half of its profit from 75% of a barrel by making low-cost transportation fuels. The other half of its profit comes from chemicals. So we see a future not where refineries are only making a single product like ethanol or a hydrocarbon from plant biomass, but they're functioning more as an integrated lignocellulosic biorefinery, making a mixture of fuels and chemicals to change the economics and to increase the deployment of this technology moving forward. So with that, I will stop, and we'll see if we have time for any questions. Thank you.
So we have time for 2 quick questions. There are no questions. Maybe I have a quick question.
Sure.
So your redesigned lignin, that zip lignin, you were talking about applying this, you've been applying it to poplar and also applying that to the field. What are the other kind of biofuel relevant plants that you are applying or are planning to apply that to?
So we are working with, I would say, 2 types of major plants. We're working with grasses in general, switchgrass. We're working with native prairie as well in terms of processing. We are also working a lot with woody biomass that processes differently than grasses. So a lot of our effort now and in the last couple of years and hopefully moving forward is going to be in cropping systems like native prairies and woody biomass.
Okay, great. Let's give Tim another hand.
Thank you.
Thank you.